Hello and welcome. Today I'm going to be going through the FRQ section for the 2022 AP Biology exam. We're going to go through all 6 of the FRQ questions. We're going to look at how to break them down, the answers, the different types of answers they can give, and the marking scheme as well. So I'm just going to jump right into it, and we're going to start off with number 1. Number 1 and number 2 are usually fairly long. You'll have 2 rather long FRQ questions that are worth about 9 to 10 marks, and then you have 4 that are a little bit shorter. Those are usually worth, I want to say like 4 to 5 marks. Okay, so these first 2 are really big and they're going to take a while, but the last 4 are relatively short. Okay, so just getting into this first one, we're going to read it, and it looks like they're giving us a diagram of what seems like some sort of signal pathway, and then we also have a table that we have to read. Okay. So I'm just going to jump right into it, and we're going to read this. It does look fairly complicated, but again, like I say, they're going to throw kind of complicated diagrams at you, and I'm going to read the paragraph, and it's going to be a little bit overwhelming, but when we actually look at the questions, they're not too bad for this one. Okay, so I'm going to start off just by reading the question and breaking it down. So it says, the binding of an extracellular ligand to a G protein coupled receptor in the plasma membrane of a cell triggers intracellular signaling, which is seen in figure 1A. So just to kind of go through what that looks like, again, so you have A here, B, and C. If we start off at A, which is our first step, you have your extracellular ligand, which is going to act as your signaling molecule, and it's going to attach to the receptor. That's the first step of any, signaling pathway. You need to receive the signal, and then we'll amplify it, and then you get the response, which is part 3. So our extracellular ligand binds to the receptor. After ligand binding, GTP replaces GDP, that is bound to GS alpha, a subunit of the G protein. So that is B. So figure 1B, you have GTP is going to turn into GDP. If you haven't heard of GTP, it's pretty much the same as ATP. It's just another energy molecule. So like in how an ATP turns into ADP when it loses its energy, it's the same kind of thing. So this is going to cause GS alpha to activate other cellular proteins, including adenylyl cyclase, that converts ATP to cyclic AMP. So now you have this GS alpha, which only became activated after you spent this GTP. Again, the GTP is an energy molecule, so it's likely you spent the GTP to make the energy to activate this GS alpha. And then what's going to happen is that GS alpha is going to turn ATP into cyclic AMP. So you've got ATP turning into cyclic AMP once the adenylyl cyclase is activated, thanks to our GS alpha. Okay. The CAMP activates protein kinases, seen in figure 1C. So this is starting to look like what our response is. Cyclic AMP is going to activate our protein kinases, make them active. And then it causes further signaling and ultimately results in the secretion of chloride ions. So this is the last step. You're going to have chloride ion secretion. So this is the whole pathway of how this happens. They give you the nice little diagram for this. And this is saying that this is under normal conditions. GS alpha hydrolyzes GTP to GDP, thus inactivating adenylyl cyclase and stopping the signal. Again, seen in figure 1A. Okay, so always read your figure captions just in case. This one says, under normal conditions, ligand binding to a G protein coupled receptor results in chloride ion transport from an intestinal cell. That's like the simplified way to explain it. You really just have ligand binding, and then through G protein coupled receptors, you're going to have chloride ion transport. So this next part of the question is giving you a little bit of context to the experiment and the table that we're going to look at. This one says, individuals infected with the bacterium Vibrio cholerae experience severe loss of water from the body, also known as dehydration. This is due to the effects of the bacterial cholera toxin that enters intestinal cells. Scientists studied the effects of cholera toxin on boar samples of isolated intestinal cell membranes containing the G protein related signal transduction components seen in figure 1. GTP was added to samples 2 and 4 only. Cholera toxin was added to samples 3 and 4 only. The scientists then measured the amount of CAMP produced by the adenylyl cyclase in each sample. So that's going to be table 1. Okay. So just by looking at table 1, you have 4 samples, and they're going to vary in whether or not they have GTP or whether they not have cholera toxin. So the minus is going to indicate that, in this case, GTP is absent, and the plus means that GTP was added. And then same things go for the cholera toxin. That negative is going to mean that there's no cholera toxin, and then these two do have cholera toxin. So just by looking at this, you can see that you have every combination of GTP and cholera toxin, which is what you want in your experiment, right? You want to do one where there's none of each, one of each, and then, both, so that you can see the difference. And what you're ultimately measuring in this case is important. It is the rate of cyclic AMP production. So we've got the metrics here of the rate that it's being produced. And again, you can see where cyclic AMP is being produced in terms of our signal transduction pathway. Okay, so I know this one seems like a lot with the diagram and everything, and we don't spend too much time on signal transduction. But you'll see that the questions for this one are pretty straightforward, at least for the first few, because they're always going to ask you some sort of, background theory question that just is based off of your knowledge of the curriculum. So those ones are usually pretty good. If you studied, then, these should be fairly easy for you. The last ones are usually the ones that are a little bit more application-based. So let's start off with the first one. Okay. So A says, describe one characteristic of a membrane that requires a channel to be present for chloride ions to passively cross the membrane. And then we'll leave the second part for after. Okay, so what's important is they're asking you to describe one characteristic. So a feature of a membrane that would require a channel protein to be present for chloride ions to passively cross the membrane. Okay, so this goes back to what we talk about actually from Unit 2, which is discussing what can and can't cross the membrane. The number one thing that has a really, really hard time and can't cross the membrane on its own are ions. Anything charged cannot cross the membrane on its own. Doesn't mean that it still can't go passively. It needs to go through facilitated diffusion. So with the help of a protein, in this case, a channel protein. So let me just draw a quick little diagram here so we can get a good idea of what we're working with. So for A, our lipid bilayer is like this. So you have your phospholipid heads and your fatty acid tails that are going to make this bilayer. And then a channel protein is quite literally going to act as a channel or a tunnel to go through the membrane. Okay. So this is a little diagram here, and then your chloride ions would need to go through, either to go in or out of the cell, but it has to go through a channel protein. It cannot pass on its own. And so they're asking you what feature of the membrane makes this basically impossible for a chloride ion to go on its own. Why does it need to go, through a channel? Okay, so there's a couple different options that you could say, but the main one is the one that we study the most. So again, what it comes down to with being able to cross the membrane is solubility. Okay, they need to be soluble. And the inside of the plasma membrane in this case, this is where the tails are. Let me grab a different color actually. These are where the tails are, and this is nonpolar, right? Because the tails are nonpolar. Right here is polar because the heads are polar, and right here is polar again because the heads are polar. So what our plasma membrane basically has is a massive region in the middle of non-polar. Okay, so for something to be able to cross the membrane, it actually has to be non-polar. I always remember like dissolves like, so polar things will dissolve other polar things. Non-polar things will dissolve other non-polar things. The cell membrane, the majority of it, is non-polar. Okay, so that means that polar ions or polar molecules in general are not going to be able to cross the membrane on their own. Yes, they can still cross the membrane, they just can't go on their own. Only small, uncharged, nonpolar molecules can, passively cross on their own without the help of any of these channel proteins. So, what I would say for this one is you're asked for one characteristic. So I think I would say for this one, I'll give you both options, that the interior of plasma membrane is nonpolar. And this would stop ions from being able to cross through on their own, hence why you need a channel protein. Okay, so they were just asking for a characteristic of the membrane that makes this basically not possible. It is because the interior of the plasma membrane is nonpolar. Only nonpolar solutes can go through without the help of a channel protein. Another thing that you could say, you could have said nonpolar / hydrophobic. Those terms you can use pretty interchangeably. So it doesn't matter which one you use, they pretty much mean the same thing. And then another option that you could do is that the interior of the membrane is also not charged. Okay, so that is also going to make it hard for a ion, something charged, to go through. Okay, so interior is also not charged. Okay. So that's it for the first part of A. You have to say either that it's nonpolar / hydrophobic, or the interior is not charged. That's why a chloride ion cannot go through on its own. Now for the second half of this, it's asking you to explain why the movement of chloride ions out of an intestinal cell leads to water loss. Okay, so this is still a general question. Obviously it pertains to what we're looking at here, but again, still fairly general. So why would the movement of chlorine out lead to water loss? Well, we need to think about our ideas of osmoregulation, right? So if I have a cell here, this, and the chlorine is moving outwards. So I've got chlorine that's moving outwards. And it leads to water loss. That means the water is also leaving. So H2O is leaving with the chloride, and they're asking why this happens. Well, this comes down to whether something is hypertonic or hypotonic, okay? And that's going to dictate the flow of water. And the flow of water is always going to be determined by the solute concentration. So in this case, chlorine is rushing out of the cell. That means that the inside of the cell is less concentrated and the outside of the cell is more concentrated, because you're pumping chlorine to the outside of the cell. So now you have low Cl inside your cell, and then you have high Cl on the outside. Our concept of osmoregularity says that what water is trying to do, and again, this occurs when you're trying to balance out the solute concentration on the inside and the outside. So when a solute, again in this case because it's an ion, can't easily cross the barrier, the easiest thing after that is to move the water. If you can't move the solute to regulate the solute concentration on the inside and the outside, then you have to move the water. So in this case, chlorine is rushing out. It's giving you a really high concentration of chlorine on the outside of the cell. Water is going to rush out as a result, okay? And that is because it's trying to stabilize, again, the solute concentration on the inside and the outside. By sending water to the outside of the cell, you are then decreasing the solute concentration on the outside and increasing the concentration on the inside as a way to regulate it. So this already has low chlorine. If I send water out of it, I'm going to increase the concentration of chlorine, right? The outside has high chlorine, so if I add more water to it, I'm diluting it and I'm bringing down that concentration to ultimately make these concentrations equal. So that is why water is flowing out of it. Water is always going to flow to where it's most concentrated. So chlorine's flowing out, that means water is going to flow out too. Again, it's always going to follow the solute because it's attempting to dilute that solute so it has an even concentration on the inside and the outside. So when water moves out of a cell, it's known as being hypertonic. Hypertonic is the cell is going to shrink because the water is moving out. Hypotonic is the opposite if water were to flow in. Okay, so what I would say for this one is going to be that the space becomes hypertonic and then water is going to move out so that it shrinks. So let's write that down here. Space outside the cell. Hypertonic. And then that will allow water to flow out. Water will flow out, again, in an attempt to dilute it, and then the cell will shrink. Okay. So that's the answer for that one. Another way that you can put this, it depends on how your teacher teaches you it. I typically teach hyper versus hypotonic, but they also might teach you it in the concept of water potential. So that's also just another way of saying the same thing. So what you would say is that the space outside of the cells now has a lower water potential compared to the cell, so compared to within the cell, and so water is going to move out. So I'll write that as well if anyone wants to choose that. It's, you know, correct as well. You would just, it's a different way of saying hypertonic. You say that the outside has a lower water potential. So hypertonic, you can also say slash, or you can also say lower water potential. Say there's a lower water potential, and then again, water is going to flow out of the cell. Okay, so those are your answers for A. It was a two-part question, 1 mark for each, the explain and then the describe one characteristic. Okay, so that one wasn't too bad. Now to get to B, it's looking like we're going to have to answer some questions about the variables. So B says, identify an independent variable in the experiment, and then we'll do the rest after. Okay, so there's, I believe, a couple different things you can say for this one. So B, we're going to first identify the independent variable. So I'll just put independent variable. Okay. So you always have an independent and then you have a dependent variable. Your independent variable is going to be typically what you're changing, and then your dependent variable is going to be what you're measuring. So your independent variable is typically your x-axis, whereas your dependent is your y. So in this case, there's actually 2 things that you could say for this one. Again, independent is going to be what you're changing, and then to ultimately see what the result is in the dependent. So the 2 independent variables are GTP and cholera toxin, because that's what we're changing, right? We're trying to see if having GTP or no GTP affects CAMP production, and then we're also trying to see if cholera and no cholera are going to change the CAMP production. In this case, CAMP would be our dependent variable. So your x-axis would have, you know, with cholera, without cholera, with GTP, without, and then your y-axis would be a bar graph probably of the rate of cyclic AMP production. So they only ask you to identify one, so you can say either. So say either presence or absence of GTP, or you can say presence or absence of cholera toxin. So I'm just going to write this very simply. So cholera versus no cholera. And then you could also write GTP versus no GTP. Okay, you didn't have to write both of them. This is only 1 mark. Either of them would work because in this case you have 2 independent variables. Okay, so there was a second part to this. It says, identify a negative control in the experiment, justify why the scientists included sample 3 as a control treatment in the experiment. Okay, so let's just start off with the first one, which is identify a negative control in the experiment. So a negative control is always going to be what we kind of consider something that's null. So you're not actually doing anything to this. The negative control is going to kind of be like a baseline. So the negative control is always going to be something where you're doing nothing to it. If you're trying to look at GTP and cholera and the effect of that, your negative control would be none of that included, okay? Because you're trying to see what it just normally is before you start adding these independent variables. It'll give you a good frame of reference. So in this case, there are a bunch of options that you could say. You could actually, in this case, you could say any of the first 3. In my personal opinion, the first one is the best because that's like the complete negative control because you're negative in both cases. But as well, these 2 are also considered negative controls because number 2 is negative in cholera toxin. This one makes it really good to compare to this one and to this one, right? So if it's positive here and negative here, you can see the effect that just the addition of cholera toxin has. So this means with GTP, it's 10. With GTP and cholera, it's 127. Okay, and same thing goes for number 3. You're looking at, again, no GTP versus having cholera toxin, and you can compare all of these. This one's like the double negative. So let me write down the whole range of options, but it's always going to be something that does not have the independent variable affecting it, so it can act kind of as a baseline. So I personally, the first one that came to my head was number 1, just because it's like the double kind of negative. So let's list the negative controls. So sample 1 is an example, which is no GTP, no cholera. Shift that down a little bit. Another thing that you could say is the ones that lack cholera, because again, those are kind of negative controls for cholera, but they still have GTP. So you could say sample 1 and 2 because they lack cholera. So 1 and 2 because they lack cholera. It's a little bit complicated because, again, you have 2 independent variables. That's why such a wide range is allowed. You typically don't have like multiple different negative controls if you only have 1 independent variable that you're changing. If you have multiple independent variables, you kind of need multiple negative controls. You could also say the sample that lacks cholera toxin but contains GTP, so that is number 2. And then you could also say the samples that lack GTP, so samples 1 and 3. So basically you could say 1, 2, 3, but not 4, because 4 is our double positive one, and then you would have to kind of explain as to why. So, sample 1 is a negative control because it doesn't have any GTP or cholera. You could say that number 2 is also a negative control because it's lacking in cholera, but it does have GTP. So that's going to be really helpful when comparing it to this one, right? Because then again, you'll see the direct cause of the cholera toxin. So for this one, you can basically say any one of these 3 as long as you justify it by saying that it's because you are lacking in one of the independent variables. You're not changing it. Okay, and then the last part of part B says, justify why the scientists included sample 3 as a control treatment in the experiment. Okay, so sample 3, we kind of already talked about as a control. Sample 3, you have no GTP and you have cholera toxin. So what this is able to do is you're going to be able to compare this to, obviously, number 4. So if I compare number 3 to number 4, this one's going to give you 0.5 and this one's going to give you 127. What's the only thing that's changing between these is GTP. So that tells you that this big difference in cyclic AMP production is again likely due to the fact that it's lacking GTP in sample 3. Okay, we're not changing both of them, we're only changing one of them. And again, the point of that is so whatever happens to your dependent variable, you know is directly caused by what you changed or what you didn't change. Okay, another thing, you could also compare it to number 1 as well. So if I'm, negative in both GTP and cholera, I'm 0.5. If I'm only negative in GTP and positive in cholera, I'm also 0.5. So that's going to also tell me something, and it's mostly going to tell me that, the cholera toxin is having no effect on cyclic AMP production in this case, right? Because the only difference between this one and this one was whether or not there was cholera. Clearly, since the CAMP production didn't change, the cholera is having no effect. If I didn't have this here, there would be nothing for me to compare it to. I would be like, okay, it's 0.5 when I have cholera and no GTP, but what am I comparing that to, right? So that's why number 3 has to be used. It's the same thing that goes for number 2 as to why. So in terms of what I would write down for these, you can use sample 3 as a control. It allows you to compare CAMP production, so our dependent variable, with that of samples that have cholera and GTP. So it's, you can compare 3 with 4. You could also compare sample 3 and sample 4, which is going to enable scientists to evaluate whether the activity of the toxin requires GTP. So that's a little bit farther, but the main point of this is going to be to really just see what that effect of that GTP is going to be. Okay. So for B, I'm going to say that sample 3 is a control. Let me move this down. Sample 3 as control, because you can compare to 4 to see direct effect of GTP. Okay. Okay. So I think that should be good. There's a couple different ways you can phrase it, but again, you want to have every single combination of these variables because you want to be able to compare them to one another. Okay. Alrighty, so what I'm going to do now is move on to C. I'm just going to erase this so that we have room. Okay, so question C says, based on the data, describe the effect of cholera toxin on the synthesis of cyclic AMP. Calculate the percent change in the rate of cyclic AMP production due to the presence of cholera toxin in sample 4 compared to sample 2. Okay, we'll start off with this first one. They're just looking for the overall effect of cholera toxin on the synthesis of CAMP. Okay. So now let's go back up to our table. This is not going to tell us anything. This is just the pathway. This is giving us, actually what's happening to the cyclic AMP production. Okay. So if we're looking at the effect of cholera toxin, we obviously need to look at a sample that does have cholera toxin and a sample that does not have cholera toxin. Okay. So if we look at, so number 1 over here has neither. We can compare this to number 3. And you can only compare this to number 3 in this case because they're both negative here, so we don't count that. However, this one's only positive in the cholera toxin. So it's showing you that because the only difference between number 1 and number 3 is the cholera toxin, whatever change in the rate of cyclic AMP, or in this case, the lack of change, is due to the cholera toxin. So because these two both have the same rate of cyclic AMP, then we can say that In this case, there is no change. So cholera toxin had no effect, right? Because I basically took number 1, I added cholera toxin and turned it into number 3, and I saw no change. The other thing that I could have said as well for my comparisons is 2 and 4. Okay, those ones can also be compared to each other, right? Because they're both positive for GTP, but number 2 is negative for the cholera toxin. So the only difference between these ones is, again, 4 has cholera toxin and 2 does not. If we look at the change in cyclic AMP, this one without cholera is 10. This one with cholera is 127. So that's quite a big difference. In the case of this, there is going to be an increase. So the cholera toxin, when you're comparing 2 and 4, causes an increase in cyclic AMP. So for this, they accepted multiple answers. It depends on which ones you compared to one another. So if you compared 1 and 3, then you would have said that there was no effect in the absence of GTP, okay? Because both of these have no GTP. So I'm going to write down the full answers for these ones. So cholera, no effect with no GTP. And I'm just going to write in brackets that we were comparing 1 versus 3. And then for the next one, cholera has an effect if you are comparing 2 and 4. So you didn't have to put both of these, you just had to put one or the other. So you can say cholera increases cyclic AMP with GTP. And that's for 2 versus 4. Okay, so either one of those you could have said. It really just depends on which ones you're comparing to each other. But you're not going to get the mark unless you specify that it's no GTP or yes GTP. Because again, you can't compare 3 and 4. I mean, you can, but it's not going to give you any information on like the cholera toxin, right? Because they're both positive for cholera. You want to look at ones that are positive for cholera and compared to ones that are negative for cholera. And obviously there's 2 different individual variables here. So you could say either one of these. It really just depended on which ones you compare. Okay, so the next part of the question says, calculate the percent change in the rate of cyclic AMP production due to the presence of cholera toxin in sample 4 compared with sample 2. Okay, so I already have these highlighted. So again, sample 2 versus sample 4, like I said, you're going to see the direct effect of the cholera toxin in the presence of GTP because they both have GTP. However, number 2 is missing the cholera toxin. Number 4 has the cholera toxin. So again, whatever the difference between 2 and 4 is, you know is caused by the fact that one has cholera and one does not. It's asking you for the percent change. You should know your percent change equation. I don't think they give it to you. But just in case, you should always remember it. It might be on your formula sheet, but just remember regardless. The formula for percent change is always going to be the same. It's going to be final minus initial over initial. And since it's a percent, you're going to multiply that by 100%. Okay, so it's for 2 and 4, and so you need to know which one's your final and which one's your initial. The final in this case is going to be number 4, right? Because we're seeing what happens when you add cholera. So the one that already has cholera added would be your final. The initial one would be prior to adding cholera. So in this case, our final value is 127 and our initial value is 10. 127 minus 10 over 10 times 100%. So this, I believe, equals 11.7 times 100%. And so your final answer should be 1,170%. 170%. So this is your final answer. That's the percent change. There's no other answer for this one. This is how you do this one. Okay. Alrighty, so the last question for this one is going to be D, which is looking a little bit longer, and this is likely going to be like our big application type of question. Now this one wasn't too bad. You didn't have to look at the like the big diagram too much. It really just was testing your knowledge on pretty much controls and what you compare to what for an experimental. Okay, so D says a drug is designed to bind to cholera toxin before it crosses the intestinal cell membrane. Scientists mix the drug with cholera toxin and then add this mixture and GTP to a sample of intestinal cell membranes. Predict the rate of cyclic AMP production in picomole per milligram adenylyl cyclase per minute if the drug binds to all the toxin. Okay, so let's just do that one. Okay, so a drug is designed to bind to cholera before it crosses the membrane. So our drug is binding to the cholera toxin. Scientists mix the drug with cholera and then add this mixture and GTP to a sample. Okay, so in this case, there is GTP, so it's asking you for the most likely number, and we're going to use our chart for this one. So basically, the GTP is positive in this case, sorry. You're adding it to GTP, so GTP is a yes. So it's going to be similar to either number 2 or number 4. So GTP, we do have. The cholera toxin, however, is being bound by a drug. So when something is bound by a drug, in this case, this is like the typical mechanism of how drugs work, it's going to be inactive. So I don't know the exact mechanism because they don't specify, but if a drug binds to the cholera toxin, it very likely will not cross the membrane or won't be active or will not be able to do its job, right? It's similar to if you bind anything to an enzyme, you're going to pretty much render it useless. So in this case, you're taking a drug, an anti-cholera drug, and binding it to cholera. So in that case, you're going to have no cholera. Okay, the drug is basically taking the cholera out of the equation, but you still have GTP. That means that you basically have the exact same conditions as sample number 2, right? With GTP, but no cholera toxin because their cholera toxin was taken out by the drug. So if they have you predict what the most likely rate of cyclic AMP production is, it's going to be 10 because these conditions are identical to our sample 2. Okay, they're a little bit different because in this case they didn't add cholera toxin, or like in the original one they didn't add cholera toxin. In the trial that we're doing for D, we did add cholera toxin, but we basically inactivated it with the drug, which is pretty much the same as not adding it at all. So for D, the most likely is going to be 10, and make sure you add your units. They're asking you specifically for these, so just make sure you add the units. So it's going to be 10 picomole per mg adenylyl cyclase per min. Okay? So that's the first part of question D. Okay, so the last part says, predict the rate of cyclic AMP production. Oops, no, I already did this. Okay. It says, in a separate experiment, scientists engineer a mutant adenylyl cyclase that cannot be activated by GS-alpha. The scientists claim that cholera toxin will not cause excessive water loss from the whole intestinal cells that contain mutant adenylyl cyclase. Justify this claim. Okay, so we're doing a separate experiment where the adenylyl cyclase is not activated. So this kind of has us going back to our diagram here to just kind of refresh our brains as to what adenylyl cyclase does. So we see it here, but it also obviously mentions it to us in the question. So let's just find this. Okay, So this GS alpha to activate other cellular proteins, including adenylyl cyclase that converts ATP to cyclic AMP. So our adenylyl cyclase, its role is to convert ATP to cyclic AMP. So what is going to then happen to, what were they asking? They're saying it will not cause excessive water loss. Okay, so I'm just going to write down my kind of thought process because this one's a little bit more of an application question. So if it's mutant adenylyl cyclase, so you have mutant adenylyl cyclase, it means it's inactive. It's just not going to work. So if my adenylyl cyclase doesn't work, like I said, I'm not going to get CAMP. So no CAMP for this one, because the enzyme that makes CAMP is not working. They're obviously, their final kind of hypothesis has to do with excessive water loss and the ability for water to be lost. So we need to go further down in the pathway. What effect does having no CAMP do? Because right now we don't know anything about how much water is leaving or not leaving. So what happens when we have no cyclic AMP? Let's go back to here. Okay, so cyclic AMP activates our protein kinases. So our cyclic AMP is going to activate these kinases. Without cyclic AMP, you're going to have no active kinases. So obviously put this in a full sentence, but I'm just writing out the pathway. No active kinases. And then finally, so the CAMP-activated protein kinases cause further signaling and ultimately results in the secretion of chloride ions, okay? So the active protein kinases causes chlorine to be secreted. We do not have any active protein kinases because we didn't have cyclic AMP because our adenylyl cyclase was inactive. So ultimately, you're gonna get no chlorine secretion, okay? So this whole pathway has been disrupted because you've mutated adenylyl cyclase. So you actually have no chlorine secretion. Okay, this is what happens. So mutant adenylyl cyclase gives us no CAMP, gives us no active kinases, and then gives us no chlorine secretion. The scientists are claiming that cholera toxin will not cause excessive water loss from whole intestinal cells if it contains this mutant adenylyl cyclase. Is this true? So if I have no chlorine secretion, like we looked at previously, I believe in part A, when you have your cell and chlorine leaves, your water is also going to follow. That's how you get water loss. So this is going to cause water loss when you do have chlorine secretion. If I have no chlorine secretion, I'm going to have no water loss because the whole point of the water being lost is because it's following the chlorine trying to dilute it. So if all the chlorine is inside, you're going to have no water loss. Okay? So this one is actually true, and you're just going to justify it using what I said here. So it says, you could say that, that cyclic AMP is not going to be produced. Okay, the protein kinases won't be activated, and then as a result, chlorine ions will not be secreted, and so you're not going to have any water leave the cell, or at least not as much water leave the cell. Okay, so that's why you get less or not as much water loss, is because you're not pumping out those chlorines due to that mutant adenylyl cyclase. Okay, so this is question 1. It was quite a long one. This one, I believe, is worth 9 points. So let me just go through. A is going to be worth 2 points, B is going to be worth 3 points, C is worth 2 points, and D is worth 2 points. So if you're marking yourself. Okay, so that is it for question 1. That was our first big one.